Assalamu alaikum warahmatullah. So continuing with our previous lecture, in this lecture we will talk about the applications of the PCR. So in this lecture we will be discussing about the various types of the PCRs. Then we will talk about detection of pathogens using PCR, of course. Then diagnose application of PCR in diagnosis of specific mutations. We'll talk about application of PCR in prenatal diagnosis. It is application in DNA fingerprinting. It is application in direct detection of microorganisms. And it is application in paleontology. And of course, it is application in diagnosis of plant pathogens. So now first, we will talk about the different types of the PCRs. Now PCRs are of several types, but the most common ones that are frequently used and commonly used are reverse transcriptase PCR. This reverse transcriptase PCR, it converts an RNA molecule first into a cDNA molecule. So RNA as we know, it is a single-stranded nucleic acid, ribonucleic acid. This single-stranded. This is first converted into cDNA or the complementary DNA molecule. So once this complementary DNA molecule is formed, then it proceeds with the normal amplification of this complementary DNA. This type of PCR is called as reverse transcriptase PCR. This is very frequently used. Number second is the real-time PCR. Real-time PCR, it processes DNA, or it is process of DNA amplification in this PCR is monitored in real times, and therefore it is used to quantitate DNA in samples. So it is again the most commonly used PCR, especially in the DNA fingerprinting and all that. Number third type of PCR is the asymmetric PCR. Asymmetric, as the name suggests, it amplifies out of the two strands of DNA only one strand. So instead of the normal or real-time PCR, which amplifies both strands, where each strand of the double-stranded DNA molecule acts as a template, here in case of asymmetric PCR, only one of the two DNA strands is being amplified. Apart from these three, there are other less frequently used PCRs like hot start PCR or nested PCR. So these three, the first three, and among first three, the first two are most commonly or most frequently used PCRs. Here is a diagram, there is a picture rather, that shows an fully automated PCR. Now let us move on to application of these PCR in detection of pathogens. Now normally or traditionally, the pathogens are detected by serological tests, wherein the presence of pathogen is detected by its antigen, or it is presence is detected by the presence of the antibody produced by our body against the pathogen. The presence of either antigen or antibody in the body confirms the presence of the pathogen. Or another traditional method is the culture method. In culture method, what we do is that we culture a given sample in a lab so as to look for the presence of the pathogen in it. But there are several pathogens that grow quite slowly. Therefore, their cells or if they are viruses, they are found in far less number in the infected cells or tissues. Hence, for their diagnosis, PCR based assays have been developed. Now, what does these PCR based assays do is that they will detect specific sequences of pathogens. Specific sequences means that they will detect specific sequences of the genome of the pathogen present in the infected cells, amplify them so as to become visible so that we are easily able to detect them. For example, these have been successfully used, PCR has been successfully used in detection of HIV or human immunodeficiency virus, which is the causative organism or causative organism of a very serious and notorious disease of human beings, that is AIDS or acquired immunodeficiency syndrome. Now the another most frequently used this test is the detection of SARS-CoV or the SARS coronavirus 2 or novel coronavirus or COVID-19, which is nowadays in most recent times, this is detected by using this PCR. So I will briefly describe how PCR is used in detection of SARS coronavirus 2 or novel coronavirus 2. Now for this, the first thing to do is that this sample is collected. Now sample is collected from the patient's nasal swab. A nasal swab of the patient is taken, as you can see from this diagram, or a throat swab is taken. Now this sample containing the sample of saliva or sometimes even a liquid from the lower respiratory tract is sometimes also taken. That sample is taken to the lab. Now once in the lab, from the sample the nucleic acids are extracted, and that nucleic acids might also contain the genome of the virus or coronavirus or SARS coronavirus 2. Now then certain specific regions of the genome of virus are amplified using reverse transcription PCR. Now why reverse transcription PCR? Because this SARS coronavirus 2, it has RNA as it is genetic material. It is an RNA virus or it is a retrovirus. So therefore, in order to amplify its genome, a specific portion of its genome, only reverse transcriptase PCR is used.